system in which every gene has been mutated. So you could just send away and get the mutated gene, and sure enough, a mutation of the PDE4 gene in C. elegans leads to the same hyperactive phenotype that you see in the schistosome parasite. And then finally, what one could do is you could take the gene from the schistosome parasite, you could graft it into C. elegans, and you could show that you could actually rescue the C. elegans mutant. So that's important for validating this as the molecular target, but now also it means that instead of having to use the big schistosome worm in your assay, at least for looking at phosphodiesterase 4 inhibitors, you could use C. elegans as your screen. It's much smaller and has a much higher throughput. So this is an example of where you start out by screening against the whole organism. You see this sort of weird phenotype, and now you can use the surrogate, C. elegans, as a way to screen at least against this specific molecular target. So that's a nice cost-effective way to kind of go through a large number of compounds. And there's a number of other specific molecular targets that are being looked at in these organisms. And these are some of them. I won't go into any detail, but if any of you have worked on any of these, you'll recognize them as having targets in other systems as well. And so that's a big part of this drug discovery for neglected diseases, is trying to pick out molecular targets that what companies would call are druggable. So what's druggable means? That means that there's a drug out there that we take that targets these types of enzymes. Okay, so you can guess, I mean, proteases, wow, you already know that. How about the HIV protease inhibitor, or any of the antihypertensive drugs like captopril targets protease. Kinases, we all know that kinases are big targets in industry now for cancer. Probably the best example being Gleevec, which is very good against a specific type of cancer in the stomach. Okay, and then 14-alpha demethylase sounds really weird, but it happens to be the target for a lot of the antifungal drugs that are effective against diseases like aspergillosis. Okay, so in all of these cases, you have examples of where there have been drugs produced, and so if you're looking at these in parasites, you've got a pretty good idea that you're probably going to be able to take advantage of that. And you can also take advantage of the fact that companies have made a lot of inhibitors of these, but you can go ahead and test, particularly the ones that weren't effective for them. That's exactly what you want. Something that might hit a parasite enzyme, not a host enzyme. So the other advantage of looking at a molecular target is you can do it computationally. And some of you may know about this approach, but I just want to make it available for everybody to know, because there's a lot of places that are doing this right now, both in academia and the industry, and it's called molecular docking. So what you do here is you start out, you have to have a structure of your target, which is true for all of those targets I just showed you with parasites. And then software has been written, and actually it was kind of interesting that this software was originally written here by Tak Kuntz many years ago, and then his postdoc, Brian Schoiket, modified it. And what it does is it takes a virtual library of compounds, and this one is called Zinc, and this stands for Is Not Commercial, and it's an open source library. If you have a laptop and you're online, you could go and look at the website, and it'll show you there's now more than 2 million compounds that are in that database. So it takes the whole database and it screens it against your target in all possible configurations. And then it gives you an output of the best scoring molecules based upon how well they fit here in the active site to activate the target, both in terms of the steric effects as well as interactions that might occur by salt bridges or other things that you know about in chemistry. And what's interesting is that the output of this, it tells you, that, let's say you pick the top 100 hits, it tells you the 100 hits, and it tells you where you can get them. And it gives you the company, it gives you the number from their catalog, and it gives you the price, right? So it's a very easy way to screen for a lot of things. Yeah.
So how well does the computer model correlate with the actual? And then the question is, why are they only, I mean, 2 million is a lot, but why is it limited by how many compounds you have? It's just any compound you can make up, I don't see why it's
Right. Okay, well, good questions. Let me take the second one first. Why only 2 million? I think it's maybe 4 million now, but why only 4 million? Okay, when you can imagine chemical space is much bigger. Okay. The reason is that this has been parsed down to compounds where you could be pretty certain that if you got a hit, you could take that into a culture model or into an animal model. Okay. So there are millions of possible chemicals out there, but most of them are greasy molecules which may be effective here, but when you try to make them into a drug, you never get anywhere because they're too insoluble. They just crash out. Okay, so those have been eliminated. And you can actually, if you go to this website, you can actually see that you can even make your window narrower. You can say, okay, only show me compounds which are related to known drugs. Okay, so you can get even more specific in terms of what you're looking for. So that's why there's so few, because you're eliminating a lot of things which could never possibly be drugs. Another reason to eliminate, what if they were things which were explosive? Or if you tried to manufacture a lot of them, you would have to have some kind of huge factory, which is just impossible to do. So all of those things go into eliminating. And then how well does it work was your other question. So one way to look at that, actually there have been some interesting publications about this, but one way to look at it is to say, if you take this molecular docking approach, which is entirely computational, and you take the top 100 hits that they give you, okay, how many of those actually are good inhibitors of the receptor or enzyme? And the bottom line is about 10%, okay, about 1 in 10. You say, well, that's not very good. Actually, that's very good if you think that you're screening through 4 million compounds in 1 week. Okay, so it really speeds things up. And then you take those ones that are actually good and you start taking those down the pipeline. So a lot of what this provides you with, and it's used by companies too, is what's called, they call it scaffold jumping. So what's a scaffold? A scaffold is like what you put up on the side of a building, but to a chemist, a scaffold is the shape of a certain molecule. So you might be dealing with one particular scaffold and you want to go and figure out something else. You can't really, in your mind, do it. You let the computer do it for you. The computer gives you a totally different scaffold and you sort of go off on that one. So scaffold jumping is a big advantage. Yeah.
Has any work been done kind of doing the opposite, where you have a target structure and looking to see what would potentially fit in that and designing a molecule that will fit a structure?
Yeah, so that's actually called rational drug design. I'll give you some examples of that in a minute. And that's actually probably, I would say, even more advantageous in this, in the sense that if you already have a structure where you've got the target and you've got a real molecule docked in there and you have the structure of that, then a good chemist can look at that and say, could I make this better by maybe putting out a fluorine here to interact with this amino acid? And that's exactly the type of iteration that allows you to go from a good compound to a very good compound. And that actually is called rational drug design. And it's based upon the ability to have the target structure co-crystallized with one of those hits.
I'm curious how you navigate some of the intellectual property challenges, because presumably some of these compounds are owned by companies or startups or things like that.
Right. So the intellectual property thing is a big issue. Okay. First of all, you would think, well, we're talking about neglected diseases, nobody's going to make any money, so why should it be an issue? The issue is that none of the partners who would sponsor a clinical trial, like the National Institutes of Health or the Gates Foundation, will do it unless they're comfortable that the intellectual property side of that compound has been dealt with. Okay? So in some cases, it really depends on the company. So in some cases, companies will just say, as long as you only use our compound to treat a human neglected disease, we'll let you do whatever you want with it and we won't get involved. We retain the rights to use that compound for veterinary disease. Now this is kind of scary because there's lots of examples where there have been compounds that have come over from the veterinary side to the human side for treating ivermectin as an example. And there's that example of the things that gone the other way. And if you think about it, companies are much more interested in what they call companion animals than they are in children in Africa. I mean, it's sad but true. I mean, little Fifi you're willing to spend $20,000 to treat your little dog, you know, but forget about it if you're talking about a kid's model. So as long as you're not going into their veterinary space, they'll say, fine, you can be eligible. So that's pretty common. Whether you do it this way or whether they donate stuff to you, which I'll tell you. On the other hand, there's some companies that'll say, no, you know, I'll go back to them and I'll say, hey, we found this, and they'll say, sorry. And the reason that they're worried is that their, they call it counsel, meaning lawyers. Their counsel told them that if we're developing one of their compounds that may be a great, you know, it's like their next blood pressure lowering drug. If we're developing it for Chagas disease in South America, we start treating 10 million people with Chagas disease. They're treating 100,000 people with high blood pressure. We're more likely to come up with some type of bad side effect, which will totally kill their market. So they don't want you to go out and start treating people in the millions. They'd rather keep it kind of close. So in those situations, some companies have said to us, we can't take it further. But there's been enough examples where companies have been pretty good about it. And recently being the last 2 years, there's been a nice push by some people like Bill Gates, for example, got together with CEOs from all the big pharmaceutical companies in London. And he basically embarrassed them. He said, what are you guys doing for neglected diseases? And he made a big splash in the paper. And he embarrassed them, and they said, okay, what should we do? What should we do? Okay, so and then suddenly I started getting a lot of things sent my way by these companies. So sometimes people can embarrass them into doing it, and they want, and especially drug companies, you might imagine, they want good publicity because of all the bad publicity they've done. So that can help with intellectual property. Okay, so this gets into your question again, in that, remember I said in the beginning that companies weren't really interested in this area, and that's true, but companies will sometimes provide you with their compound libraries for screening against these organisms for the reasons I said, that there's something called, some of you may know about this, very important for CTSI, it's called a material transfer agreement. It's the agreement that's signed off by UCSF or UC, Office of the President, Contracts and Grants, with a company saying that, okay, we're receiving this library of 10,000 small molecules, and we agree to screen them only against this, and if we find something, it's our responsibility to take it forward, and we're not going to go into your area of veterinary medicine. So it's all written out. It's called material transfer agreement. So all of these companies have given compounds to UCSF under material transfer agreements. Big companies and small companies, and companies that don't exist anymore. So, you know, variety. And so this is actually good because since companies are in the process of making drugs and they want to do this as easily as they can, they often will have already vetted their libraries in animals. So you know that what they're sending you is safe to give to mice because they've already looked at that, which is not true necessarily of compounds that you might get from another academic chemist, for example. So one example is this particular compound, which came from Solera most recently. And it's very effective against the organism Trypanosoma cruzi that produces Chagas disease. Anybody who took I3 probably seen this. This is a picture of Carlos Chagas, and this is actually the life cycle of this is the life cycle of the parasite right here. It's spread to you by this thing called a kissing bug, and it infects your heart. And it's the leading cause of heart disease in Latin America. And we were just talking about this from the question in the back about this so-called rational drug design. So here's an example of how it's done. So in the back here, this is a surface representation of a target. This is a protease from that organism. Stick figure here is the first of that scaffold, that molecule that was provided by Solera. And then this medicinal chemist, Bill Rausch, who's at Scripps Research, Florida, made a series of derivatives. And each of those derivatives was then co-crystallized with the target so that you could actually see how it's interacting in different ways with the target. And it turns out that in this particular derivative, you had a very nice interaction with this green space here, which is hydrophobic, and this is hydrophobic. So this actually increased the efficacy and selectivity of this compound series for this particular target. So this is exactly what we were talking about. This is this co-crystallization and so-called rational drug design, where a chemist and a crystallographer will work together to redesign, literally, redesign molecules to make them more effective. Okay? And so you keep doing this, rounds and rounds and rounds of this until you kind of hit the wall and you say, okay, I can't come up with anything that's better than this. This is as good as I can get. And at that point, then you start a second process of making that compound drug-like, that is, making it orally bioavailable, having a long half-life, etc. Okay. So this compound had to go through this screen that we talked about to show that it actually it's parasites. And then it has to go into a mouse model. And this is one screening mouse model that you should be aware of. It's one that's used. Well, there's actually a machine that does this over in the Cancer Research Institute. There's also one that's in a BSL-2 level for parasites. But basically what you do is you have a parasite which has to be genetically modified. It's genetically modified, so it's carrying the gene for luciferase. Okay, so you basically have a parasite that at least is compatible with putting in an important gene. And then you inject mice, and you can see, you can actually externally visualize the replication of these parasites. So this is 3 days, 6 days, 8 days. Now you see the parasites are all over the mouse. Okay? And you can do that in the control, and then you can have a series of mice in which you treated them with a particular drug. This happens to be one of these antifungal drugs. And you can see you completely clear parasites from the mice. And a nice thing here is you don't have to kill the mice. This is done by external visualization of this luminescent output. So this is the type of screen that you use to go through these compounds. Or you can do a longer screen in which you actually look at heart failure, which is what these guys produce in mice. And you can also do this in dogs. And I mention this because one of the things that came out from this dog study is that a particular cardiac enzyme called troponin I that some of you may be familiar with because it's used to indicate in clinical laboratories the size of a myocardial infarct. It also turns out to spike up in dogs that have been infected with Trypanosoma cruzi because this parasite kills heart muscle cells. Okay? So if you do that and then you treat these dogs with that particular compound, you find that you actually prevent this troponin I spiking. And the reason this is important is now troponin I is being looked at in Brazil as a possible biomarker to see response to therapy in patients with Chagas disease. So that's important in terms of a clinical trial. Okay, so coming back to this pipeline, I've told you a lot about all these different approaches and screens, and yet everything I've told you has only gotten us here. Okay, so target family is selection, and all this chemistry that we talked about, whether it be done computationally or done by rational drug design, only gets you to here. So how do you do all the rest of this stuff, right? And I'll just briefly tell you that the whole preclinical testing, which has to be done in, remember, non-rodents, right, has been done by what's called a contract research organization. Another very important thing to know in CTSI, because right now there's an attempt to put together a series of vendors, CRO vendors by CTSI, so that people at UCSF who come up with drugs and get to this point of preclinical development can talk with CROs about getting this kind of stuff done. Okay? So what is this stuff? Formulation. So what is formulation? Okay, we don't know about drugs. Some of you may know what formulation is, but formulation is basically what form is that chemical now going to be when it's given to people, right? Is it going to be a tablet? a gelatin capsule, a solution, powder in a bottle. Okay, so there's a lot of different possibilities. It turned out for this protease inhibitor, it was bitter. It had to be sweetened. Okay, that's part of formulation. It has to taste good. Safety studies in dogs and monkeys. This refers to absorption, distribution, metabolism, excretion. You have to know all of that for something that you're giving to dogs and then you can give it to people. And then you have to make a whole bunch of the stuff. Okay, so manufacturing is very important. And all of this is done by these contract research organizations. The one that's used primarily here is SRI International, which is right down in Menlo Park. Okay? But the key is to do this for just one compound. One compound costs about $2 million. So here's the thing. We're talking, if this was a company, hey, no problem. A couple $1 million, we can do that easily. But really, the people who have come through for neglected diseases that any of you who work in these areas could now go back to is actually the National Institutes of Health. Of course, not today because the government shut down and they're not working, but often they'll do it. And they actually have a contract with SRI International to get this kind of stuff done. And so they probably, more than anyone else, really pay for this. Okay? Some of the early development, some other foundations that pay, but they're really the ones who pay for it. And I'll just end with one story about one that I think you'll find interesting, because it kind of tells you how you can get this kind of stuff done. So we were trying to get this protease inhibitor into these preclinical tests, because once you have those done, then you go to the FDA and you go to people. And so we went to this guy, well, these two guys, Phil Coyne and John Rogers, who were at NIAID, National Institutes of Allergy and Infectious Disease, and said, hey, you know, we got this thing. They said, oh yeah, we know about that. It's really great. You know, we said, well, we're trying to get this now into preclinical testing, but it costs a couple $1 million. And they said, well, we do have a contract with SRI, but would you mind talking to this other woman? I said, no, I'll talk to anybody. So they had me talk to Beth Spinelli. So Beth Spinelli said, oh, that's great. You're trying to do this for Chagas disease, big problem in Latin America. It's emerging question in the States. We understand its importance. She said, well, by the way, does this compound have any effect against any of these diseases? And she pulled out a list of the biodefense organisms. Okay? And it just turned out that we had just collaborated with some guys up in McGill for cryptosporidium. Now you think cryptosporidium, that's a bioterrorism organism? Well, it's on the list. Okay? And we said, yeah, cryptosporidium. She said, thank you. And suddenly, the all got done. So what had happened was the NIH, you may know, got this huge amount of money from Congress directly during the reign of President Cheney. I'm sorry, because he was really paranoid about anthrax. So he got Congress to give the NIH a whole bunch of money for biodefense. And so they have all this money for drug development, but of course, who's doing it? Nobody. I mean, if the company's doing it, they're staying in the company. Nobody's working on the really bad diseases anyway. And so they were worried they were going to lose this money. So here we came along with this thing, you know, and we said, oh, it just happens to this. So she said, thank you. And we piggybacked on the biodefense contract, and we're continuing to do that. So there's another thing to think about, that sometimes when you're dealing with neglected disease, when you don't have a company backing you up, you sometimes can find some other clever way to get that thing funded. So I think this is a good place to stop, because it'll leave us with a couple of minutes if you guys have questions. Yeah.
How willing are countries like China willing to fund some of this, especially since they have more money now?
Right. So there are countries who are willing to fund manufacture and are willing to support clinical trials. It's interesting, most governments are not willing to invest in early discovery research. Okay? I think because the failure rate is so high that they don't want to get involved in that. But if you're down the line, so for example, for Chagas disease, one of the reasons we picked it is there are countries in South America, Brazil, Argentina, Colombia, Venezuela, who have the manufacturing capacity to make the drug distributed, and there are ongoing clinical trials that you can check. So that works, and for China, schistosomiasis works because it's a big problem there, especially with all the dams that are going on the Yangtze now. And so they're willing to manufacture and they're willing to support clinical trials. But none of this stuff before. So you have to get something up to that point.
I think I remember hearing from, I don't remember where, like a speech about how oncopsoriasis is one of the big success stories, where I don't remember what company where that was able to come with or made it very available.
Right, so onchocerciasis alias river blindness. There is a drug that was donated by Merck called ivermectin, which kills the microflare, which are little guys that crawl around. And so for transmission, it's great. And that drug came from the veterinary world. It turned out to kill parasites that were in cow dung. That's why it was the term cattle. So Merck is still donating ivermectin to the WHO. The problem is it kills the microflare but doesn't kill the adult worms. So now there's a big move to try to go with what's called macro flarecide, something that kills the adult worms, because as long as the adult worms are there, when you stop treating with ivermectin, the adult worms just produce more. So the Gates Foundation is very much into this research now to try to come up with the new macro flares.
Other than like shaming and embarrassment by big public figures, what are the other incentives and companies that they're going to get their libraries?
Well, there's no economic incentive. Okay, so it all comes down to it's not shaming, it's good publicity. And now there are organizations who are trying to pull things out of companies. One of them is called WIPO, World Intellectual Property Organization, and it's associated with another organization called BioVentures for Global Health. And what they do is they try to broker interactions between companies and people doing these kinds of neglected disease screens. And they just say to the companies, look, you don't have anything to lose here, and if something comes up that's good, you can have the publicity. So it really comes down to that, because unfortunately there's no economic incentive. And people always say, well, what about malaria? You know, like every time I go to Costa Rica, I have to take Malarial or something. Well, companies have actually done the economics of that, and they just won't make enough money. Even with ecotourism, even with the military, you don't make enough money. At best, it's like cost neutral. So that's not gonna work. So it's all about this. Yes.
Is vaccine development for these kind of, was using the same three methods that you've described?
There is a lot of effort going on for vaccine development, because obviously if you had a good vaccine for any of these diseases, that would be a way to control it. And malaria and schistosomiasis, there's been vaccine development attempts, discovery and development attempts going on since the 1970s. Nothing. So I always tell, when I give the lecture of my eye through class, I say, yeah, we're going to have a vaccine for malaria about a week after we have the vaccine for HIV. You know, I mean, it's the same issue. I mean, these organisms are so highly involved in how to evade the immune response. And often, when you induce an immune response, you actually make the disease worse. And that happened with hookworm. There was a hookworm vaccine that actually got to the point of being tested in people. And what happened was, as they got infected with hookworm, there was this huge immune response in their skin, and they got this terrible rash, and they just had to stop. So it's really hard. I mean, there aren't any now, and I don't see any on the horizon. That's no problem. So I think we can probably stop.